Lesson 1: Terminologies and Forms of Fish. Fish preservation. Fish preservation is any operation that can prevent or inhibit the natural process of breakdown or decomposition taking place in the fish. It is known as fish processing. So, fish preservation involves various methods to prevent or inhibit the natural breakdown or decomposition of fish, collectively known as fish processing. These methods, including refrigeration, freezing, drying, smoking, canning, salting, and pickling, aim to extend the shelf life of fish by controlling bacterial growth, enzymatic reaction, and moisture content. Terminologies in fish preservation. First, autolysis. It refers to the softening of tissue in fish, which occurs due to the digestive action of enzymes. This process is a result of the fish own enzymes breaking down its tissues. After that, it is important in the context of fish preservation to understand how autolysis can affect the quality of the fish. Next is brine. In fish preservation, brine is commonly used as a solution to soak or immerse fish. The salt in the brine helps to preserve the fish by drawing out moisture and inhibiting the growth of spoilage, causing microorganisms. Third is can sealer. In the context of fish preservations, cans are often used as containers for fish products, and the can sealers ensures that the can is tightly sealed to maintain the freshness and quality of the fish inside. Fourth is canning. Canning is a method of preparing food, including fish, in a hermetically sealed container. The process involves placing the food in the can or jar, sealing it tightly, and then applying heat to destroy microorganisms that could spoil the food. Canning is an effective way to preserve fish for an extended period. Fifth is dehydration. In fish preservation, dehydration can be employed to extend the shelf life of fish by reducing the water content, which inhibits the growth of bacteria and molds that cause spoilage. Six is drying. Drying is a preservation method that involves lowering the moisture content of fish through the application of natural or artificial heat. This process inhibits the growth of microorganisms that thrive in moist environments, extending the shelf life of the fish. Dried fish is lightweight, has a longer storage life, and is a common method in various cultures. Next is eviscerate. Evisceration refers to the removal of internal organs from the fish. This process is essential in fish preservation to eliminate organs that can contribute to the breakdown or decomposition of the fish. Evisceration is commonly performed to prepare fish for further processing or specific preservation methods. Next is exhausting. Exhausting is the elimination of air inside the can during the canning process. Removing air is crucial to create a vacuum seal, preventing the growth of aerobic microorganisms that require oxygen. This airtight environment helps maintain the quality and safety of the fish stored in the can. Gibing is the process of removing internal organs from the fish by making a triangular cut at the throat. This method is often associated with preserving certain types of fish, such as herring. Gibing helps in preventing the release of enzymes that can lead to the deterioration of fish quality. Next is icing. Icing involves the application of ice to lower the temperature of the fish. This method is commonly used for short-term preservation, especially during transportation. By reducing the temperature, icing slows down bacterial growth and enzymatic reactions, preserving the freshness of the fish. Next is pH. pH refers to the hydrogen ion concentration in a solution. In the context of fish preservation, monitoring pH levels is crucial to understanding the acidity or alkalinity of the environment. Maintaining the right pH is important in pickling processes, where the acidity of the solution helps inhibit microbial growth and contributes to the distinctive flavor of the preserved fish. Next is pickling. Pickling is the process of curing fish in a solution of vinegar and spices. This method imparts a unique flavor to the fish while creating an acidic environment that inhibits microbial growth. The combination of vinegar and salt and spices contributes to both the preservation and taste of pickled fish. Pressure cooker. A pressure cooker is a closed metal container equipped with a pressure gauge, control bulb, and petcock. While not exclusive to fish preservation, it can be used in certain canning processes. The high pressure inside the cooker raises the boiling point of water, allowing for faster and more efficient heat penetration during the canning process. Pressure gauge. A pressure gauge is a device used to measure and maintain pressure during processing, such as in canning, and ensure that the pressure inside the canning environment remains at the desired level and contributing to the safety and effectiveness of preservation process. Next is refrigerator. A refrigerator is a method of preservation that involves lowering the temperature to a point where bacterial growth is inhibited, while refrigeration is commonly associated with short-term storage and it plays a crucial role in preserving the freshness of fish by slowing down enzymatic reaction and microbial growth. Next is rigor mortis. Rigor mortis refers to the stiffening of the muscles or tissues of a fish or any organism several hours after death, and it occurs due to chemical changes in the muscles leading to a temporary state of rigidity. Salting. Salting is a method of preserving fish in which salt is the main preservative. This process involves covering the fish with salt, which helps to draw out moisture and create an environment unsuitable for the growth of spoilage, causing microorganisms. Next is smoking. Smoking is a preservation process for fish that involves the application of smoke along with other interrelated processes such as salting, drying, and heating treatment. The smoke not only imparts flavor but also acts as a preservative, inhibiting bacterial growth. Next is vacuum. Vacuum refers to an empty space without air. In the context of fish preservation, vacuum packaging is a technique where air is removed from the packaging to create a vacuum. This helps in preventing oxidation and spoilage, extending the shelf life of the fish. Last is viscera. Viscera refers to the internal organs or entrails of a fish. When processing fish for consumption, the removal of viscera is a common practice to ensure that only the edible portions are retained. Market forms and cut of fish. First is whole or round fish. Whole or round fish refers to the structure of a fish where all parts are intact. This means that the fish has not been filleted or cut into pieces and it is presented in its entirety. When you purchase a whole or round fish, you get the entire fish as it is, including the head, tails, fins, and internal organs. This type of presentation is common in market and seafood stores, allowing customers to see the complete form of the fish before preparation. It's worth noting that preparing a whole fish may involve cleaning, scaling, and gutting the fish before cooking. Next is drawn fish. Drawn fish involves the removal of internal organs by creating a slit along the belly wall and pulling out the entrails. This process is performed to clean and prepare the fish for consumption, making it ready for cooking by eliminating non-edible parts. Next is dressed fish. It is refers to the form of fish where scales, inner organs, fins, head, and tail are removed, leaving only the edible portion. This preparation is commonly done for convenience, allowing for easier cooking and consumption without the presence of non-edible parts. Next is sticks. Fish sticks are thick slices of fish, typically cut perpendicular to the muscle fibers with a thickness ranging from 1 to 2 centimeters. These sections provide a convenient and uniform way to cook fish, allowing for even cooking and presentation. Next is splitted. The term splitted refers to the form of fish that is often along the dorsal side from head to tail. This preparation allows for easy access to the internal portion of the fish and is often done for specific culinary or processing purposes. Next is live fish. It is refers to the form of fish that is provided or sold while still alive. This is often done to ensure the freshness of the fish, and it allows customers to choose live fish for specific culinary preferences or cultural practices, which live fish may be prepared immediately before consumption. Next is butterfly fillet. It is a type of fish fillet where the fillet are joined together on the ventral side through the removal of the underside skin. This technique is often used in cooking to create a visual appealing presentation and to allow for more even cooking. The term butterfly refers to the way the fillet are opened up and joined together, resembling the wings of a butterfly. This method is commonly used for flat fish like flounder or sole. Next is single fillet. A single fillet is the portion of fish meat that is cut away from one side of the fish, typically separated from the backbone. This fillet is boneless, making it convenient for cooking and consumption as it eliminates the need to deal with bones. Next is sticks. Sticks is usually referred to cuts made from minced and shaved fish. They are often breaded and frozen for convenience. Next is common body forms of fish. First, fusiform. Fusiform describe a torpedo-like physique shape that is streamlined and tapered at both ends, facilitating swift and efficient swimming in aquatic environments. An example of a fish with a fusiform body shape is the galunggong, also known as round scad or mackerel scad, which exhibit this adaptation for enhanced aquatic mobility. Next is compressiform. Compressiforms refers to a body shape that is flat and compressed laterally, meaning it is broad from side to side. An example of a fish exhibiting compressiform body shape is the slip mouth, showcasing a flatted and laterally compressed structure that is adapted to its specific ecological niche. Next is depressiform. It is describes a body shape in which the dorsal side is depressed vertically, creating a flattened appearance. An example of a fish exhibiting depressiform body shape is the stingray, characterized by its flattened body with a vertically depressed dorsal fin, which aids in its bottom-dwelling lifestyle. Next is tiniform. It is used to describe a ribbon-like body shape in fish. An example of a fish exhibiting tiniform body shape is the hairtail, which features a long and slender body resembling a ribbon. This adaptation allows for streamlined movement through the water and is often seen in species that navigate often aquatic environments. Next is anguilliform. It is described as serpentine or eel-like physique shape characterized by a long cylindrical body. Eels exemplified by species like the common eel showcase this adaptation, allowing them to navigate through water with a sinuous, snake-like movement. And lastly is globiform. It is refers to a round and globular physique shape and an example of a fish exhibiting this body shape is the popper. Poppers are known for their ability to inflate into a ball-like form as a defense mechanism, showcasing their round and globular adaptation. Lesson 2: Fish Preservation Principles and Agents of Spoilage. Principle of fish preservation. First, prevent or delay microbial decomposition. Microorganisms such as bacterial, fungi, and molds can lead to the decay of fish. To prevent this, measures like refrigeration, freezing, or the use of preservatives can be employed to slow down or inhibited microbial growth. Next is prevent or delay self-decomposition. Fish undergo natural enzymatic activities that can contribute to their self-decomposition. Rapid spoilage can be avoided by promptly cooling or freezing the fish after catch to slow down these enzymatic processes. Last is prevent other causes of spoilage, including physical damage. Physical damage, such as brushing or crushing, can accelerate the spoilage of fish. Careful handling and proper storage methods, like using appropriate packaging and avoiding rough transportation, help prevent physical damage and maintain the fish quality. Changes in fish after death. First, changes in appearance. As spoilage processes, the color of the fish start to fade until it becomes dull. This change in appearance is an indication of the deterioration of the fish. The fading color is often a result of chemical and enzymatic reaction taking place in the fish tissue. Second is rigor mortis. It is refers to the stiffening of the muscle tissue in the fish. This state is a natural process that occurs after the death of the fish. It is typically lasts for a few minutes in some fish to several hours in others. Fish in the rigor mortis state is considered fresh and its duration can vary among different fish species. Third is autolysis. Autolysis is the breakdown of tissue caused by enzymes present in the fish. This enzymatic activity softens the tissue and provides a conductive environment for the growth of microorganisms. Autolysis contributes to the overall deterioration of the fish, and controlling it is crucial for preserving fish quality. And lastly is the putrefactions. It is the state when the components of the flesh break down into assimilable forms. This stage is accompanied by a foul odor, which results from the breakdown of proteins in the fish. Fish at the putrefaction stage is considered spoiled and undesirable for consumption. In summary, these stages illustrate the progressive deterioration of fish from changes in appearance to the development of rigor mortis, autolysis, and ultimately putrefaction. Proper fish preservation method, as mentioned earlier, play a vital role in slowing down or preventing these stages, ensuring the fish remains safe and suitable for consumption. Characteristic of fish and stalefish. Eyes: bright, bulging pupils velvet, black corneas transparent. Dull weakened sunken pupils, dull black corneas opaque. Next is gills: bright red, covered with clear slimes, fresh odor under gill cover. Dull brown or gray slime, cloudy, odor under gill cover, sour and offensive. Next is flesh: firm, stiff body, impression made by finger do not remain, soft and flabby, impression made by fingers remain. Belly walls: intact, often ruptured viscera protruding. Next is muscle tissue: white, pinkish, especially around backbone. Vent: pink, not producing, pale brown producing, odor, fresh, fishy odor, stale, sour or putrid, color, bright, faded, and dull. Agents of spoilage. First, microorganism. Three main groups of microorganism. First is bacteria. Bacteria play a vital role in breaking down organic matter, such as fish, after its death. Their activities result in processes like spoilage, fermentation, and decay, contributing to the return of simple inorganic compounds to the soil and recycling of nutrients in ecosystems. As fish is an organic material, it is susceptible to rapid spoilage once bacteria start acting upon it after death. Next, molds. Molds thrive in environments with moisture, warmth, and organic matter. They play a crucial role in the natural decomposition of organic materials, but their presence of food can lead to spoilage and potential health risks. To prevent mold growth, it's important to store food properly, keep areas clean, and address any moisture issues. Next is yeast. Yeast are microorganisms that form wheates to yellowish colonies on the surface of food, often presenting as a film on liquids or as sediments at the bottom. Their presence can contribute to various fermentation processes in food, influencing taste, texture, and aroma. And the second one is enzymes. Enzymes are proteins that serve as biochemical catalysts in living organisms, aiding in the acceleration of chemical reactions. Typically present along the digestive tract, enzymes play a crucial role in breaking down complex food molecules into forms that can be easily assimilated by the body, facilitating nutrient absorption and utilization. Classification of bacteria according to shape. First, coccus, round. Second, bacillus, rod shape. Third, spirillus, spiral. Fourth, staphylococcus, grape-like. Fifth, streptococcus, chain-like. Classification of bacteria according to their temperature requirement. First, thermophile, thrive at high temperature or heat-loving. Second, psychrophilic, thrive best at low temperature or cold-loving organisms. Third, mesophilic, those that thrive between low and high temperatures. Classification of bacteria according to their oxygen requirement. First, aerobic, requires free oxygen in order to live. Second, anaerobic, lives in the absence of free oxygen. Third, facultative, grows with or without free oxygen. Types of enzymes. First, lipolytic, enzyme that on fats. Second, proteolytic, enzymes that acts on proteins. Third, amylolytic, enzyme that acts on carbohydrates.